Welcome to the Explainer. So glad you're joining us today because we're opening up something I find absolutely fascinating, the Molecular Detectives Toolkit. We are going to look closely at the incredible variations of the polymerase chain reaction, or PCR. If you're ready to see exactly how scientists decode some of the most complex genetic mysteries out there, you are definitely in the right place. Now, right off the bat, we got to ask ourselves a crucial question. Traditional PCR, it's literally a molecular miracle. It is fantastic at making millions of copies of a specific DNA sequence. But what happens when it's just not enough? Think of standard PCR like a hammer. It's incredibly effective, but sometimes you don't need a hammer. Sometimes you need a scalpel or a magnifying glass or a multi-tool. Today, we're exploring exactly what scientists do when the standard hammer just won't cut it. Here's a quick roadmap for today's explainer. We'll start by looking at going beyond traditional PCR. Then, we're unpacking 5 specialized tools: pinpointing exact mutations, multitasking with single strands, detecting RNA viruses, boosting sensitivity, and finally, exploring entirely unknown DNA sequences. Let's get right into it. Okay, part 1. Beyond traditional PCR, building out that toolkit. You see, as scientists pushed the limits, trying to answer wildly complex biological questions, they inevitably hit roadblocks. The standard PCR just couldn't tell them everything they needed to know. So, innovation took 2 distinct paths. First, some techniques became highly specific optimizations of the traditional method, basically tweaking the original recipe. The second path, completely novel creations designed from the ground up for really unique lab challenges. Which brings us to section 2, pinpointing exact genetic mutations. So how do we find a change in just a single DNA building block out of literally billions? Well, the answer is ARMS. That stands for Amplification Refractory Mutation System, also known as allele-specific PCR. Think of ARMS as a highly precise molecular scalpel. It's built specifically to hunt down SNPs, single nucleotide polymorphisms, just one single altered building block. And the whole system operates on a brilliant, simple rule. The primers only attach, or anneal, if their 3' end perfectly complements the exact target sequence. And what's so incredibly cool about this mechanism is the definitive yes or no answer it gives us. If that specific primer perfectly matches the target at that crucial 3' end, boom, the Taq polymerase enzymes do their thing, they extend the sequence, and we get our product. But if there is even one tiny mismatch right at the end, Taq polymerase hits a brick wall. 0 extension. It's a completely definitive test for whether a specific mutation is there or not. Moving right along to section 3, multitasking and single strands. Let's say you only need one strand of DNA for a test. We use a technique called asymmetric PCR. In standard PCR, you mix in equal amounts of forward and reverse primers, right? You get your standard double-stranded DNA. But here's the trick with asymmetric PCR. You intentionally skew the recipe. You put in a ton of one primer and hardly any of the other. Why do we do this? Because getting single-stranded DNA, or ssDNA, is absolutely essential when you're preparing hybridization probes. Here's how it actually plays out. Initially, the abundant primer and the limited reverse primer work together, making regular double-stranded DNA. But pretty soon, that limited primer just runs out. It's totally consumed. Once it's gone, the abundant primer, which is still in excess, just keeps right on working solo, churning out copy after copy of our desired single-stranded DNA. It is so clever. Now, what if you want to look for multiple targets at the exact same time to save precious lab hours? Enter multiplex PCR. This is the ultimate laboratory multi-tool. Instead of running just one test per tube, you toss multiple primer sets into the exact same reaction. Obviously, you save a ton of time and money doing this. The catch, however, is that you have to make sure the amplicon sizes, the lengths of the DNA pieces you're creating, are distinctly different from one another. That way, they show up as completely separate bands later during visualization. One quick heads up, though, a trade-off for all this efficiency is that the sensitivity could be slightly lower than standard PCR. But the sheer efficiency here is just staggering. Imagine combining 4 separate strains into one single reaction instead of running 4 completely different tubes. You end up with beautifully distinct bands, say at 371, 275, 202, and 141. It's elegant, it's incredibly fast, and it is a massive time saver for diagnostic labs. All right, section 4, detecting RNA viruses. Let's look at a massive real-world problem. Standard PCR only reads DNA. So, how on earth do we track and study RNA viruses like HIV or hepatitis C? We use reverse transcription PCR. It's an absolute genius workaround. Step 1, you take your RNA template. Step 2, since normal PCR enzymes are blind to RNA, we introduce a special enzyme called reverse transcriptase. This literally translates the RNA into a complementary DNA format, known as cDNA. Once you have that cDNA, step 3 is simply running your standard PCR amplification with your specific primers. It is hands down the most sensitive method out there for RNA detection. In practice, you've got two main ways to pull this off. You can do a rapid one-step method, tossing the RNA, the reverse transcriptase, and the DNA polymerase all into a single tube, which is great for keeping contamination risks low. Or you can use the gold standard two-step method. You make the cDNA first, and then deliberately transfer it to a separate tube for the specific amplification, giving you way tighter control over the whole process. Let's dive into Section 5, boosting PCR sensitivity. If your DNA target is incredibly scarce, it's highly prone to nonspecific binding, which basically means frustrating lab errors. We have to boost our sensitivity. To fix this, scientists came up with nested PCR. Think of it as a two-layered filter. Instead of one round of PCR, you do two. Round 1, you use primers to do a broad sweep, generating a large outer PCR product. Then, in round 2, you take that product and use a completely different set of nested primers. These are designed to bind precisely inside that first product, generating a much shorter, highly specific inner target sequence. And the reason this is so brilliant is perfectly summed up by this quote from our source material. It is very unlikely that any of the nonspecific PCR products contain binding sites for both primers. Think about it. If round 1 accidentally amplifies a piece of random junk DNA, the odds that this junk DNA also contains the exact binding sites for your second inner nested primers, practically zero. It is an incredibly clever way to eliminate false positives. And finally, Section 6, exploring unknown DNA sequences. Imagine trying to amplify a DNA sequence when you only know a tiny, tiny fraction of the code around it. Sounds impossible, right? Well, scientists use inverse PCR. It involves a crazy physical trick. Step 1, you isolate a restriction fragment of DNA. This has your small, known sequence and the big unknown part you want to read. Step 2, you force that fragment to undergo self-ligation. You literally make its ends glue together, forcing it to do molecular acrobatics into a closed circle. Step 3, once it's a circle, you synthesize primers from the known part to amplify the unknown part. This whole mechanism is a total molecular mind bender. By forcing that fragment to self-ligate, the DNA loop essentially turns inside out. The primers bind to the known region, sure, but instead of pointing inward at each other like normal, they point outward. They run along the circular track and amplify all that completely unknown genetic territory. It's such a beautifully creative way to map uncharted genetics. And that wraps up our dive into the Molecular Detectives Toolkit, from using arms to catch a single mutated nucleotide, to translating RNA viruses into readable DNA, to literally bending DNA into inside-out circles with inverse PCR. The sheer versatility of these techniques is mind-blowing. But with biotechnology advancing at such a breakneck speed, it leaves us with one incredibly thrilling question to think about. What future variations of PCR are scientists going to invent next? Thanks for joining me on this explainer. Keep asking those big questions, and we'll catch you on the next deep dive.